This is Lucian Williams, test 3.2, and today I'll be discussing RNA versus CRISPR. So today we're using a plate as a cell, toothpicks as the RNA we're adding, grape as the protein we're adding. Together they make the vector complex. Spaghetti will stand in as our DNA, with blue tape as the sequence we're targeting, pink string as the mutations in the DNA, and string as the mRNA. So today we're stepping into the shoes of a geneticist with a problem on our hands. We would love to study the effect that underproducing a specific protein would have on a human cell. However, when we log on to our favorite cell culture website, we can't find a cell line available with the proper gene knocked out. So we're going to have to put on our thinking caps and figure out how to get around this problem using one of two methods. First up is a method you've undoubtedly heard of before, the CRISPR method. By introducing a combination of DNA-cutting protein called Cas9 and a segment of short RNA whose sequence matches our protein's gene, we can modify any normal cell we might already have on hand to disable the gene responsible for making the protein we wish to study. The complex that forms will identify a sequence of our DNA that matches the RNA and snip it. The DNA will attempt to repair itself, but in the process will likely mutate the gene, thus disabling it. Propagating the cell would then make a lineage that lacks production of the protein we wish to study and would then be a reasonable candidate. Secondly, we have the RNAi technique. In this case, we would introduce siRNA or miRNA molecules into the cell that match a sequence found in the mRNA that codes for a protein. The interfering RNA creates risk complexes with proteins in the cell, which recognize and cleave mRNA at sites that match the RNAi's sequence. Instead of targeting the gene itself like CRISPR, this inhibits protein expression by attacking the mRNA that the gene transcribes. Through this technique, we have a culture of unmutated cells that lack or minimally express the protein we are studying and thus are also candidates. For the interests of our study, I would prefer the RNAi technique. Now, both techniques have a degree of uncertainty, as their RNA components combine and affect portions of non-target molecules with similar code. The side effects of CRISPR, however, can be more detrimental to our study, as our modified cell can have unwanted mutations that make further mutation more likely. The cell line we create from this cell could then have a non-uniform genetic code and result in greater incidences of cancer, confounding our results. RNAi side effects can also be hard or impossible to detect, but should at least present consistently during testing, making them easier to account for.